Good afternoon, student. Today we are going to discuss the hybridization technique. Okay, in the last lecture we have discussed the concept of hybridization, then history of hybridization. Yes, the concept of hybridization. The hybridization, it is nothing but the crossing or the mating of the two plants or the lines of the dissimilar genotype. Okay, that is whenever the two plants having the different genetic constitution are crossed with each other, that process is called as the hybridization. Okay, that is, the process is very important as due to the use of pure line selection in the self pollinated crops for the improvement, the genetic variability in the self pollinated crop get exhausted. Okay, and the genetic variation, it is the basis of the crop improvement. Okay, therefore the genetic variability, it is to be artificially created, and for this one the plant breeder uses hybridization method. Okay, we have discussed the concept of hybridization in the last lecture in detail. Then also we have discussed the history of the hybridization. Then the objectives of the hybridization. The principal objective of the hybridization is the creation of variation, and apart from this one, the combination breeding, transgressive breeding, and the production of hybrid varieties, these are the other objectives. Okay, then also we have discussed the types of hybridization based on the taxonomic relationship of parents. Okay, now the how actually the hybridization technique it is carried out in the field. Today we are going to discuss regarding the hybridization technique. Okay, here crossing or the mating of the two plants having the dissimilar genotype, it is carried out by the seven step. Okay, it is carried out in the seven steps. Okay, the first step is the choice of parents, the second one is the evaluation of parent, the third one it is the emasculation, fourth one bagging, fifth one tagging, sixth one pollination, and the last one is the harvesting and storing the F1 seeds. Okay, hybridization technique, it is a seven step process. In this one, the first step is the choice of parent, the second step it is the evaluation of parents, then third one, after evaluation of parents, emasculation is carried out. After emasculation, bagging is carried out. After bagging, the tagging is carried out. After tagging, pollination is carried out. And the last step in the hybridization technique is the harvesting and storing the F1 seeds. Okay, these are the different steps in the hybridization. Okay, in this one, the first one, the choice of the parents. Okay, choice of the parents, generally the parents which are to be selected for the crossing or the mating, these are depending upon the objective of the hybridization program. Okay, there are different objective of the hybridization program we already discussed, and apart from these objectives, the increase in the yield, it is always an objective of the plant breeder. Okay, the selection of parents or the choice of the parent, it mainly depends on the objective of the program. In addition to the other objectives, the increase in the yield always the objective of the plant breeder. Therefore, at least, at least one of the parent should be well adapted and proven in the area for which the new variety is to be released, while the another parent must have the characters which are absent in the well proven variety. Okay, therefore generally the selection of the parent is carried out in a such way that the one of the parent, it should be well adapted and proven variety in the area where the hybridization program is to be carried out or where the new variety is to be released, while the other parent should have the characters which are completely absent in the वेल प्रूव्हन व्हरायटी. ठीक आहे, लक्षात आलं? म्हणजे काय, तर याच्यापैकी दोन जे पॅरेंट आपल्याला सिलेक्ट करायचे, त्याच्यापैकी एक पॅरेंट हा ज्या एरियामध्ये तुम्हाला नवीन व्हरायटी रिलीज करायची आहे त्या एरियामधली ती व्हरायटी ती एकदम वेल प्रूव्हन आणि त्या एरियाच्या क्लायमॅटिक कंडिशनला अडॅप्टेड असलेली पाहिजे, त्याचा परफॉर्मन्स माहीत असला पाहिजे. ठीक आहे. आणि दुसरी जी व्हरायटी आहे त्या दुसरी व्हरायटीमध्ये मात्र असे कॅरेक्टर्स पाहिजे जे की ह्या वेल प्रूव्हन व्हरायटीमध्ये नाही आहेत. ओके, म्हणजे त्याचं जेनेटिक कॉन्स्टिट्यूशन हे वेगळं हवं. ओके. देन द then later on the one should remember that the character absent in the original parents will generally not appear in the segregating generations. Okay, the character absent in the parents will not appear in segregating generations except the transgressive segregation and some complementary gene actions. Okay, some complementary gene actions, for example the some gene interactions. Thus the choice of parent, it is the basic step in the hybridization program. Okay, the choice of the parent is basic step in the hybridization program, and often more than anything else success and failure of the program. Okay, that means the success and failure of the hybridization program, it is completely depending upon the selection or the choice of the parent. Characters which are absent in the parent will not appear in segregating generations except transgressive segregation and some complementary gene action. Okay, therefore it is the considered as the most basic step, and the success and the failure of the hybridization program, it is completely depending upon the choice or the selection of the parent. Okay, that means here you can see the character which is sought to be improved. Okay, that means if suppose that the plant breeder wish to improve the disease resistance of the variety, that disease resistance capacity, it should be present in one or the other parent. Okay, remember that the character sought to be improved must be present in one or the other parent, and the characters which are absent in the parent will not appear in segregating generations except transgressive segregation and some complementary gene action. ओके, म्हणजे तुम्हाला त्या व्हरायटीचं जे कॅरेक्टर इम्प्रूव्ह करायचं आहे ते कॅरेक्टर दोन्हीपैकी एका पॅरेंटमध्ये प्रेझेंट असणं आवश्यक आहे. जर दोन्हीमध्ये पण ते कॅरेक्टर प्रेझेंट नसेल तर ते त्याच्या पुढच्या F1 F2 सॅग्रिगेटिंग जनरेशनमध्ये ते कॅरेक्टर प्रोड्यूस होत नसतं. ओके, दॅट्स वाय द कॅरेक्टर विच इज सॉट टू बी improved must be present in one or the other parent. Okay, that is all about the selection or the choice of parent. Then the second step is the evaluation of the parent. Okay, evaluation of parents means what? If the performance of the parent, if the performance of the parent in the area where the breeding is to be carried out is known, then the evaluation of the parent is not necessary. But if their performance is not known for that particular area, then the parents should be evaluated for the characters. ओके, लक्षात आलं. म्हणजे ज्या एरियामध्ये आपल्याला हायब्रिडायझेशन प्रोग्राम कॅरी करायचा आहे, समजा औरंगाबाद आहे, तर त्या औरंगाबादमध्ये जर तुम्हाला हा प्रोग्राम कंडक्ट करायचा असेल तर तुम्हाला काय करावं लागेल? जर त्या चा परफॉर्मन्स त्या एरियासाठी जर माहीत असेल तर त्याचं इव्हॅल्युएशन करण्याची काहीच आवश्यकता नाही. पण जर परफॉर्मन्स माहीत नसेल तर त्या एरियामध्ये त्याचा परफॉर्मन्स तुम्हाला इव्हॅल्युएट करावा लागतो, स्पेशली त्या कॅरेक्टरसाठी जे तुम्हाला इम्प्रूव्ह करायचं असतं. Okay, generally the evaluation or the choice of the parent, it is carried out in area where the breeding is to be done for the characters they are expected to contribute and the disease resistance. ओके, लक्षात आलं. म्हणजे त्या एरियामध्ये त्या एन्व्हायरमेंटल कंडिशनमध्ये ते कॅरेक्टर्स एक्सप्रेस होतात का आणि त्याची डिसीज रेजिस्टन्स कॅपॅसिटी आहे का, ह्या दोन गोष्टी त्या इव्हॅल्युएट कराव्या लागतात. Okay, that is the importance of the evaluation of the parents. That is, if the performance of the parents for the area where the hybridization program is to be carried out is known, then the evaluation or their testing is not necessary. But if their performance for that particular area it is not known, then the parents should be evaluated for their performance. Okay, and especially the evaluation it is carried out in the area where the breeding is to be done for the characters they are expected to contribute and for the disease resistance. Then also these parents are also evaluated for the mechanical mixtures and the heterozygosity. Okay, these are also evaluated for the mechanical mixture. Mechanical mixture means the seeds of these parents may get mixed with the seeds of the other variety or the weed seeds. That's why these parents should be evaluated for the mechanical mixtures and also for the heterozygosity. Okay, that is, we have discussed the hybridization means it is just the crossing of two pure lines having the different genetic constitution. Okay, that's why the parent should be homozygous. If the parent which is selected for the crossing it shows about 5% of cross pollination, then that parent it is considered as the heterozygous. ओके, म्हणजे जवळपास 5% पर्यंत जर त्याच्यामध्ये क्रॉस पॉलिनेशन होत असेल त्या पॅरेंटमध्ये, तर ते हेट्रोझायगस कन्सिडर केलं जातं. अँड देन लॅटर ऑन टू इंड्यूस द होमोझायगॉसिटी, these parents are then subjected to the self pollination for one or more generation to reduce the heterozygosity. Okay, understand that. That is, these parents are evaluated for the mechanical mixtures and the heterozygosity. Okay, if in one of the parent the cross pollination it increases beyond the 5%, then that parent it is considered as the heterozygous, and that parent then subjected to the self pollination for one or more generation to reduce the heterozygosity and to increase the homozygosity in the parent. ओके, दॅट इज ऑल अबाउट द इव्हॅल्युएशन ऑफ पॅरेंट. ठीक आहे, लक्षात आलं. जे हायब्रिडायझेशन मध्ये जे पॅरेंट असतात ते होमोझायगस असतात. ओके, पण समजा त्या पॅरेंटमध्ये जर 5%च्या पेक्षा जर जास्त क्रॉस पॉलिनेशन होत असेल तर ते पॅरेंट हेट्रोझायगस कन्सिडर केलं जातं आणि त्याच्यामध्ये हेट्रोझायगॉसिटी कमी करण्यासाठी त्याच्यामध्ये क्रॉस पॉलिनेशन प्रिव्हेंट करून त्याच्यामध्ये सेल्फ पॉलिनेशन केलं जातं 1 किंवा 2 जनरेशनसाठी, ज्याच्यामुळं हेट्रोझायगॉसिटी कमी होऊन जाते आणि ते पॅरेंट्स होमोझायगस होतात. ओके, that is all about the evaluation of the parent. Okay, that is all about the evaluation of the parent. Now we are discussing the third most important and critical step in the hybridization, and that is called as the emasculation. Okay, the third step in the hybridization is the emasculation. What is meant by emasculation? Emasculation, it is nothing but the removal of stamens, the removal of stamens or the anthers, removal of stamens or the anthers, or killing the pollen grains without affecting female reproductive organ gynoecium is called as the emasculation. ओके, म्हणजे काय करायचं याच्यामध्ये, त्या जे पॅरेंट तुम्ही फिमेल पॅरेंट म्हणून यूज करणार आहात त्याच्यामधलं त्या स्टेमेन्स रिमूव्ह केले जातात किंवा अँथर्स रिमूव्ह केले जातात किंवा त्याच्या पोलन ग्रेनला मारून टाकलं जातं, गायनोशियमला कुठल्याही प्रकारची इंजुरी न करता. त्या प्रोसेसला आपण इमॅस्क्युलेशन म्हणत असतो. ओके, इमॅस्क्युलेशन, द रिमूव्हल ऑफ स्टॅमेन्स or the anthers or killing the pollen grains without affecting in any way the female reproductive organ gynoecium is called as the emasculation. And what is the purpose of emasculation? The purpose of the emasculation is to prevent self pollination in the female parent. Okay, is to prevent the self pollination in the female parent. Okay, because the plant breeder have selected the another parent, male parent, from which the pollen grains are collected, and these pollen grains are then placed on the stigma of the female parent. And that's why, to prevent the self pollination in the female parents, the stamens are removed, or anthers are removed, or the pollen grains are killed without affecting the female reproductive organ gynosium. लक्षात आलं? म्हणजे आपण प्लांट बिल्डरने जे सिलेक्ट केलेले आहे मेल पॅरेंट आणि फिमेल पॅरेंट, त्या मेल पॅरेंटचे पोलन ग्रेन्स कलेक्ट करून ते फिमेल पॅरेंटच्या फ्लॉवर मधल्या स्टिग्मावरती टाकायचे. ठीक आहे. आणि त्याचं सेल्फ पॉलिनेशन त्याच्यासाठी होऊ द्यायचं नाहीये. त्याचा मेन पर्पज हा असा असतो. ओके, द पर्पज ऑफ द इमॅस्क्युलेशन इज टू प्रिव्हेंट pollination in the female parent. Okay, then generally emasculation in the dioecious plants. Okay, in the mm, dioecious plants, generally the in the dioecious plants, male plants, in the dioecious plants means what? There is the male flowers and the female flower, these are present on the different plants. Okay, in that case, simply the male plants, these are removed from the field. Okay, it is very simple in case of dioecious plant. It is also simple in the monoecious plants. Monoecious plants like in castor or the maize, you can see the male flowers and the female flowers are different. Okay, but these are present on the same plant. In case of this one, this is also very simple. Emasculation is very simple because here it is essential to only remove the male flowers. Okay, or the male inflorescences are removed in the monoecious plants. Okay, but it is a very important and essential factor in the bisexual flowers. Okay, bisexual flowers, because the male reproductive organ anther and the female reproductive organ gynosium, both these are present in the same flower, and that's why the emasculation, it is essential in the bisexual flower. Okay, don't forget that in the dioecious plant, the male plants and female plants are different. In that case, only the male plants are removed from the field. Okay, in case of monoecious plant, male flower and the female flowers, these are present on the same plant. For example, castor and maize, in that case only the male inflorescence, male flowers are removed. But emasculation is very essential in the bisexual flowers. Okay, then there are various methods of the emasculation are determined. Okay, there are large number of the methods which are used to remove the anther or the stamens or to kill the pollen grains. Okay, there are various methods, but the method which is suitable for the species, for the suitable for that particular parent, it should be determined by the size of the flower, then amount of seeds which are required, then number of seeds per fruit set, and the purpose for which the hybrid seeds are required. Okay, various methods of the emasculation. For example, number one is the hand emasculation, or simply it is called as the forceps or the scissor method. Then another method is the suction method of emasculation, then hot water treatment, alcohol treatment, cold treatment, and also the genetic emasculation. Okay, these are the different methods of the emasculation, but the method which is suitable for the particular parent, it should be determined by the size of the flower. Okay, for example, simply we can say, whenever the size of the flower, it is large sized, then simply we can we can carry out the hand or forceps or the scissor. Okay, but the flower is small, then you cannot use the hand emasculation method. Okay, that's why we are saying the suitable method for the species determined by the size of the flower, then amount of seeds needed. Okay, if you if the plant breeder need the large number of the seed, then it is completely impractical to use the hand emasculation method. In that case, the other methods the plant breeder use. Okay, that's why we are saying the best method, it is determined by the size of the flower, the amount of seeds needed, then number of seeds per fruit set, and lastly the purpose or for which the hybrid seeds are required. Okay, in the examination the separate short note it may be asked on the emasculation. Okay, in this one you have to define the emasculation, the basic concept of emasculation, and then the different methods of the emasculation. Okay, emasculation, here the first method you can see the hand emasculation method, which is also referred as the forceps or the scissor method. Okay, generally just we have discussed that the hand emasculation method, it is used, it can be used in the species which are having the larger sized flowers. Okay, anthers and the stamens, these are removed with the forceps and the scissor. Okay, for example in the wheat, barley or the cotton, and that's why this method it is also referred as the forceps scissor method. Okay, the exact procedure, the exact procedure how exactly the anthers or the stamens are removed, this exact procedure it differs from crop to crop. Okay, generally the emasculation it is done before the dehiscence of anthers. ओके, म्हणजे अँथरचा डेहिसन्स होऊन त्याच्यातले पोलन ग्रेन बाहेर पडायच्या अगोदर हे इमॅस्क्युलेशन केलं जातं. ओके, जनरली द इमॅस्क्युलेशन इज डन बिफोर द डेहिसन्स ऑफ अँथर्स ऑर द स्टिग्मा हॅज बिकम रिसेप्टिव्ह. म्हणजे स्टिग्मा ते पोलन ग्रेन रिसीव्ह करण्यासाठी तयार झालेला आहे त्याच्या अगोदरच जनरली हे अँथर्स केले जातात. Okay, the emasculation it is generally done before the dehiscence of anther or before the anther matures or stigma has become receptive. Okay, usually the plant breeder observe that the stigma receptivity it is at peak in the morning hours when the flowers opens. Okay, the stigma receptivity it is at peak in the morning when the flower open. ओके, जनरली स्टिग्माची रिसेप्टिव्हिटी कधी जास्त चांगली असते तर मॉर्निंग मध्ये जेव्हा फ्लावर फ्लावरचं ओपनिंग होतं किंवा जेव्हा फुल उमलतं तेव्हा. ओके, त्याच्यामुळं काय देअर फोर द इमॅस्क्युलेशन इट इज डन इन द इव्हिनिंग. Emasculation it is done in the evening between 4 to 6 p.m. one day before the anthers are expected to dehisce or mature and stigma is likely to become receptive. ओके, लक्षात आलं, म्हणजे अँथर मॅच्युअर व्हायच्या किंवा अँथर फुटायच्या आणि स्टिग्मा रिसेप्टिव्हिटी हाय व्हायच्या एक दिवस आधी अगोदरच्या संध्याकाळी 4 ते 6च्या दरम्यान जनरली हे इमॅस्क्युलेशन केलं जातं. Okay, therefore the emasculation it is done in the evening between 4 p.m. to 6 p.m. one day before the anthers are expected to dehisce or mature and the stigma it is likely to become receptive. Okay, and therefore the flowers which are selected for the emasculation are those that are likely open in the next morning. ओके, म्हणजे सिम्पली लक्षात काय ठेवायचंय, आपल्याला असं वाटतंय फुलाकडे बघितल्यानंतर की जे दुसऱ्या दिवशी सकाळी उमलणार आहे तर त्याच दिवशी संध्याकाळी उमलायच्या एक दिवस अगोदर संध्याकाळीच हे इमॅस्क्युलेशन केलं जातं किंवा त्याचे अँथर्स आणि stamens remove केले जातात. Okay, therefore the flowers these are selected for the emasculation are those that are likely to open in the next morning. Okay, sure, care should be taken to remove all the anthers for the flower. Okay, the all the anthers, all the stamens from the flower should be removed without breaking them, and the most important thing the gynoecium must not be injured. ओके, लक्षात आलं, म्हणजे त्यांना ब्रेक न करता हळूवारपणे ते अँथर्स हे रिमूव्ह केले पाहिजे आणि पॅरलेल मध्ये त्याच्या गायनोशियमला कुठली इंजुरी सुद्धा झाली नाही पाहिजे. ओके, in the species which are having the small flowers, in the species which are having the small sized flowers. In that case, the hand emasculation method, it is very tiring. It is very tiring, it is very difficult, and it is time consuming. Okay, and similarly, that is whenever the plant breeder requires the large quantities of the seeds, then it is completely impractical to use the hand emasculation method in the parents. ओके, लक्षात आलं. म्हणजे जेव्हा स्मॉल साईज्ड फ्लावर मध्ये जर यूज करायचं असेल तर काय, त्याच्यातले ते अँतर सोडकणं, त्याला व्यवस्थित हळूवारपणे कट करणं, हे काय एकदम डिफिकल्ट टास्क आहे, टाइम कन्झ्युमिंग आहे. ओके, आणि जेव्हा तो खूप सारे सीड्स हवे असतील प्लांट बिल्डरला, तेव्हा प्रत्येक फ्लॉवरला जाऊन हे असं रिमूव्ह करणं हे कम्प्लिटली इम्प्रॅक्टिकल आहे. Okay, that's why this method, it cannot be used in the small sized flower, and also when the large number of the seeds are needed. But for accurate genetic studies, generally the hand emasculation method, these are used for accurate genetic studies. It is desirable because in all other methods there may be some amount of self pollination takes place. ओके, जेव्हा अॅक्युरेट जेनेटिक स्टडी करायचे असतात तेव्हा मात्र हीच मेथड यूज केली जाते, कारण ही परफेक्ट मेथड आहे, ह्याच्यामध्ये कुठल्याही प्रकारे सेल्फ पॉलिनेशन होत नाही. परंतु ज्या बाकीच्या मेथड्स आहेत, सक्शन मेथड, हॉट वॉटर ट्रीटमेंट, कोल्ड वॉटर ट्रीटमेंट, यामध्ये मात्र थोड्या का होईना प्रमाणामध्ये सेल्फ पॉलिनेशन होत असतं. ओके, दॅट्स वाय द हँड इमॅस्क्युलेशन मेथड, इट इज युज्ड फॉर द अॅक्युरेट जेनेटिक स्टडीज. Okay, then the second method of the which is used for the emasculation is the suction method. Suction method, that is hand emasculation method, just we have discussed, it cannot be used in the small sized flowers, and therefore the another method was discovered, and this is called as the suction method. Okay, suction method, it is useful in case of small flowers where the hand emasculation, it is not possible. Okay, generally the process, it is carried out in the morning just before or immediately after opening of the flower. ओके, म्हणजे सकाळी सकाळी जेव्हा फ्लावर ओपन होणार आहे त्यावेळेला ही मेथड यूज केली जाते. ओके, द फर्स्ट ऑफ ऑल द petals, these are removed with the help of the forceps. Okay, first of all the petals, these are removed with the help of the forceps. Then a thin rubber tube or glass tube which is attached to the suction hose. Okay, here you can see it is a thin glass tube or the rubber tube which is attached to the suction hose, that is which is producing the suction pressure. Okay, a thin rubber tube or the glass tube, it is attached to the suction hose. It is used to suck the pollen grains from its surface. Okay, it is used to suck the pollen grains from its surface. Here you can see, here the pollen grains are sucked in this one tube, and these are collected in the this flask. Okay, but the care, it must be taken that the suction should be enough to suck the stamen and pollen grains but not the gynoecium. Okay, the suction pressure, it should be enough to suck only the pollen grains. Okay, and not the gynoecium should not be injured. Okay, in this method, the considerable amount, that is about 15% of the cell pollination may takes place. ओके, जवळपास 15% सेल्फ पॉलिनेशनचे चान्सेस या मेथडमध्ये असतात. Considerable amount, about 15% of the self pollination may takes place in this method. That means the self pollination cannot be completely eliminated in the suction method. Okay, the chances of self pollination cannot be eliminated in the suction method. Okay, that is all about the suction method. Okay, suction method. Then the another method, it is called as the hot water emasculation. hot water emasculation. Here it is a well-known fact that the pollen grains or the male reproductive organs, these are more sensitive than the female reproductive parts to the environmental and the genetic factors. Okay, the pollen grains, these are more sensitive, or we can say the male reproductive organs, these are more sensitive than the female reproductive organs to the environmental and the genetic factors. And this property, it is utilized to kill the pollen grains without affecting the female reproductive organs. ओके, लक्षात आलं. पोलन ग्रेन्स हे फिमेल रिप्रोडक्टिव्ह ऑर्गन स्टिग्मा किंवा गायनोशियम पेक्षा हे एन्व्हायरमेंटल फॅक्टर्स किंवा जेनेटिक फॅक्टर्सला खूप सेन्सिटिव्ह असतात. ओके, म्हणजे फॉर एक्झाम्पल काय, तर पोलन ग्रेन्स हे हॉट वॉटरला किंवा कोल्ड वॉटरला किंवा अल्कोहोल ट्रीटमेंटला हे जास्त सेन्सिटिव्ह असतात त्या फिमेल रिप्रोडक्टिव्ह ऑर्गनपेक्षा. आणि हीच प्रॉपर्टी इमॅस्क्युलेशन मेथडमध्ये यूज केली जाते. Okay, here you can see the pollen grains are more sensitive to the hot water than the female reproductive organs. ओके, म्हणजे जर समजा इथे तुम्हाला दिसतंय 45 डिग्री टेम्परेचरचं वॉटर यूज केलेलं आहे, तर काय 45 डिग्री सेंटीग्रेडला पोलन ग्रेन्स सेन्सिटिव्ह असतात, पण त्याचा इफेक्ट त्या हॉट वॉटरचा गायनोशियम किंवा फिमेल रिप्रोडक्टिव्ह ऑर्गनवरती होत नाही. Okay, the pollen grains are more sensitive to the hot water than the female reproductive organ. Okay, here the most important thing is the temperature and the duration of the treatment. These are important and vary from crop to crop and must be determined for the every species. Okay, म्हणजे काय, किती टेम्परेचर पाहिजे त्या पोलन ग्रेन्सला मारायला आणि त्या टेम्परेचरचं वॉटर त्या गायनोशियम वरती निगेटिव्ह इफेक्ट करणार नाही. ओके, तर टेम्परेचर ऑफ द वॉटर आणि दुसरं काय, तर किती वेळासाठी ती ट्रीटमेंट द्यायची आहे, हे प्रत्येक क्रॉप टू क्रॉप हे बदलत जातं. आणि हायब्रिडायझेशन प्रोग्राम मध्ये सगळ्यात पहिल्यांदा पॅरेंट्ससाठी हे टेम्परेचर आणि ड्युरेशन ऑफ द ट्रीटमेंट हे डिटरमाइन करावं लागतं. Okay, here the temperature and the duration of the treatment, these are important and vary from crop to crop and must be determined for the every species. Okay, it must be determined for the every species. Okay, in this treatment you can see the hot water, it is carried in the thermos flask, and the whole spike or the whole that male flowers or that anthers, the whole spike, it is immersed in the water. Here you can see the whole spike, it is immersed in the water. Okay, that is generally the treatment, it is given when the anther dehisces and prior to opening of the flowers. ओके, फ्लावर ओपन व्हायच्या अगोदर किंवा अँथर मॅच्युअर झाल्यानंतर डेहिस व्हायच्या अगोदर हे हॉट वॉटर मध्ये बुडवले जातात. ओके, जनरली द ट्रीटमेंट इट इज गिवन when the anthers dehisces and prior to opening of the flower. Okay, that is all about the hot water emasculation. Then the other methods which are used for the emasculation is the cold treatment and the alcohol treatment. Okay, cold treatment like hot water treatment, the cold treatment, or we can say the cold water, it can also kill the pollen grains without damaging the gynosium. Okay, like the hot water, the cold water, it also kill the pollen grains without damaging the gynosium. Okay, but this cold water treatment, it is less effective than that of the hot water. Why it is less effective? Because the amount of self fertilization or the self pollination, it is greater than the heat treatment in the cold treatment. ओके, म्हणजे हॉट वॉटर ट्रीटमेंट पेक्षा सुद्धा कोल्ड वॉटर ट्रीटमेंट मध्ये सेल्फ पॉलिनेशनचे चान्सेस जास्त असतात, तेव्हा कोल्ड वॉटर ट्रीटमेंट ही हॉट वॉटर ट्रीटमेंट पेक्षा जास्त इफेक्टिव्ह नाहीये. Okay, cold treatment like the hot water treatment, the cold treatment can also kill the pollen grains without damaging the gynosium. Okay, but this treatment, it is less effective than the hot water treatment because the amount of self pollination is greater in the cold water treatment than the hot water treatment. Okay, then the another treatment which is used is the alcohol treatment. Okay, generally this alcohol treatment method, it is not a very popular method. Okay, alcohol treatment, it is not generally used for the emasculation of the female parent. Okay, in this treatment a particular concentration of alcohol is used for a fixed time. Okay, it is used for a fixed time period to kill the pollen grains. Okay, that means a suitable concentration of alcohol, it is taken in the thermosflask, that flower, it is then immersed in the alcohol for a fixed time, okay, to kill the pollen grains. Okay, here also the duration of the treatment and the concentration of alcohol is of utmost importance. Okay, understand that duration of the treatment and the concentration of the alcohol, it is also of utmost important. Okay, that is all about the hot water treatment, cold water treatment, and alcohol treatment. Then the last method which is used for the emasculation is the genetic emasculation. What is meant by genetic emasculation? The genetic or the cytoplasmic male sterility may be used to eliminate the necessity of the emasculation. So the genetic or the cytoplasmic male sterility may be used to eliminate the necessity of the emasculation. Okay, generally in some of the plants the self incompatibility may be found. Self incompatibility means what? That is, the in the same flower the anthers are present and male female reproductive organ gynoecium is present, but the anthers are not compatible, so that the self pollination get avoided. Okay, then these are male sterile plants, that is these are naturally emasculated. Okay, male sterile plants are, these are naturally emasculated, there is no need to emasculate them artificially. Okay, then in certain genotypes the male sterility, it can be induced by the gene manipulation in cytoplasmic or the nuclear genome. ओके, म्हणजे काय त्याच्यामध्ये मेल स्टेरिलिटी इंड्यूस केली जाते त्याच्या जिनोम मध्ये काही मॅनिप्युलेशन्स करून. ओके, त्याच्यामुळे काय होतं, पुन्हा नंतर कुठल्याही प्रकारचं इमॅस्क्युलेशन करण्याची आवश्यकता राहत नाही. In such plant, then there is no need to use the other methods of emasculation. Okay, then the genetic emasculation, or we can say the self incompatibility or the male sterility, it can be induced by spraying the chemicals such as 2,4-D naphthalene acetic acid, malic hydrazide, and triiodobenzoic acid. Okay, these are the different chemicals which are used for to induce the male sterility in the female parent. Okay, that is all about the emasculation methods. Okay, today we have discussed the three steps in the hybridization. Okay, the first one is the choice of the parent or selection of parents, second one is the evaluation of the parent, and the third step, it is the emasculation. Okay, in the examination the emasculation, it may be asked for the short note. Okay, there are different methods of emasculation. The first one we have discussed, the hand emasculation. Hand emasculation, it is also referred as the scissor or forceps method. Then second one, suction method. Third one, hot water treatment. Then fourth one, cold water treatment. Fifth one, alcohol treatment, and the last one is the genetic emasculation. Okay, I think the time is running out. This is enough for today's lecture, and we will discuss the other steps of hybridization in the next lecture. Okay.